Hello and welcome back to educator.com. Today we're going to talk about Mendelian genetics and foundational experiments of molecular biology. Now this is going to be an interesting unit because we're talking mostly about the history of how we started with Gregor Johann Mendel, and we're going to go all the way up until we are somewhat in the 20th century at least. Now, we have to start any talk about molecular biology by starting off with Gregor Mendel. We will then go through the chromosomal theory of inheritance, and then a large chunk of this talk is going to be on the discovery of the genetic material of the cell, which we now know is DNA. We'll talk briefly on the discovery of RNA, and we will mention how the genetic code was cracked. So as I said, talking about molecular biology cannot be done without first talking on Gregor Johann Mendel. Now, Gregor Mendel lived in the 19th century, and at the time he was considered a biologist and a botanist, right? He did most of his work on plants. He published his seminal paper in 1866 on hybridization and inheritance of the pea plant. Now, unfortunately for Mendel, his results were criticized at the time of publication as only being about hybridization and not being about inheritance. And unfortunately for him, he wasn't given the respect that he deserved in his time because his results weren't widely accepted until after he passed away. And that happened in about 1900 is when his work was picked back up by other scientists. However, looking back after the fact now, we consider Gregor Johann Mendel as the father of modern genetics. Now, he did all of this work while being an Augustinian friar, so he was a monk, and this was his research that he did on the side. So, let's talk about Gregor Mendel's laws. Now, he made two very important laws based off of his research. This is what he postulated. He showed that there was a principle of independent segregation, and we're talking about alleles. Then, his second law is the principle of independent assortment, speaking toward genes. So, his first law, he started with true breeding lines for certain observable traits. So he used these pea plants, and what a true breeding line means is that they're homozygous. All right, so they always produce the same phenotype. All right, so if it's a green pea, then when you mate two green peas together, you get only green peas. Okay, now he inferred that an individual's phenotype was determined by a pair of unit factors, right? And we call those nowadays, we call unit factors, we call these genes. And he said different versions of a single gene are called alleles. Okay, so they can vary just slightly, maybe even as small as a single nucleotide difference in an entire many thousand base pair gene. And he said that alleles can be dominant or recessive. Now, what that means is that any recessive gene, for example, if we say that green, let's say, is dominant, but you can also be yellow, let's say. Okay, so green, if these are the two alleles, okay, anything, if green is dominant, then anything with two dominant alleles, big G's, will be green. Okay, anything with even one dominant allele, the big G, will be green. And only something with two recessive alleles, okay, will be a different phenotype. In this case, let's say it's a red. Right? So Your observable phenotype, which is these, is what is called a phenotype. And your genetic composition, okay, which is this stuff, that would be your genotype. All right? Good. Now, genotypes can be experimentally determined by mating and then analyzing the progeny. Okay, so you analyze the phenotypes of the progeny. Remember, phenotypes is anything you can see. Genotypes is the actual genetic makeup. So individual alleles, he says, Mendel, will segregate independently into gametes or sex cells like sperm and eggs, with each gamete receiving one allele from each pair. Okay, so one allele from each pair of alleles, right? Now, using a Punnett square, draw out all possible genotypes and phenotypes of a cross between true breeding white flowers and true breeding purple flowers, with purple being the dominant. Proceed through the F2 generation. Now, first of all, we need to know what all this means. Okay? First of all, a Punnett square is a way to visualize those matings, right? And F2 generation is what's called the filial 2 generation. So you start with your, say, parents here. Okay, mom and dad. Okay, they cross and produce, let's say, you and your sister. Okay, right here, let's say you have a baby of your own, right? When you meet your, for my example, let's say a wife. Okay, together you produce a baby. So this is what's considered the parental generation. This is the first filial generation, or F1, and this is the F2 generation. So proceed through there using a Punnett square, and remember, our Punnett squares would be looking like an actual square based on all of the genotypes that we get for a specific gene. So let's try this solution. So purple's dominant, right? Well, if purple is dominant, let's write that there. Before we start on this, let's go back to Mendel. All right? Mendel, he mated plants, right? He mated pea plants. And he looked at their seed coat and seed color. So if we do an example of Mendel's seed color or seed coat, then maybe we'll be able to understand how to do the flowers. So he had true breeding lines, meaning they're homozygous. So he took true breeding round seeds, okay, and true breeding wrinkled seeds. Okay, the round seeds are big R, big R. The wrinkled seeds are little r, little r. Okay, this is parental. Okay, when you mate them, you get all big R, little r. And remember, if round at this point is dominant, all of these will be round. Okay, now what you're going to do, you mate the F1 generation to itself. Okay, so it's going to be another big R, little r. Okay, and what you can do right? These are going to be all the gametes possible. Okay, so if these are the female gametes, okay, and these are the male gametes, right? You can make a Punnett square. And that Punnett square is going to look as such. Okay, you're going to have your females and your male gametes. All right. Now, all you have to do is fill in the box across from each other. So, this one, we have a little r, right, crossing with a big R. So this is big R, little r. These are two big R's. This right here is a big R and a little r. And this right here is two little r's. In this case, and this, by the way, is our F2 generation. So in this case, what we have here, remember, dominant is round. So we have this is round, this is round, this is round, and here's our wrinkled one. Okay? And as we see here, we have a 3:1 ratio of dominant to recessive for phenotype. Okay, so we have two different phenotypes, but we have three different genotypes, right? Because we have two of these. Two of those. Okay, so maybe this will help us get our flower one better, all right? So do we want to try? Let's go ahead and give this a try. So we have our purple and we have our white. We normally call our genes for the dominant. So we'll call our purple big P, big P. And we'll call our white little p, little p. Right? And so this is purple. All right. So when they mate, what do we get? We get big P, little p, right? And that is still going to be what color? Purple. All right, and this is our F1. All right, that was our parental. So what do we have to do? We need to mate this to another P and P, right? So what are our options? We have our little P, big P, big P, little p, right? And if, say, this is the mom and this is the dad. Okay? So if we follow this out, we can make our nice Punnett square. With P, big P, little p. So we have big P, big P. We have big P, little p. We have big P, little p, and little p, little p. So what do we have here? We have this being purple, this being purple, this being purple, and this one is our lone white. So just the same as this, we have a 3:1 in favor of purple for phenotype. And we have, once again, three different genotypes. P, 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 little p, and little p, little p. Okay, and remember we have two of these. All right, so hopefully we found that helpful. Now, what's very important is that, as I've said before, we always find the phenotype of the heterozygote, okay, or the phenotype of the dominant allele being seen in the heterozygote. So these ones, right? we're seeing the dominant phenotype in those, okay, in the heterozygote. Now, for many alleles, this doesn't always happen. Okay, we don't necessarily have to have alleles that are completely dominant or completely recessive. And Mendel got really lucky in the fact that his pea plants were true breeding lines in this way, and they were either dominant or recessive alleles. Sometimes you can have something called incomplete dominance or codominance. And that's where, let's say, for example, you had a red flower and a white flower, and you mated those. And instead of getting, let's say if red was dominant, instead of getting 3 red flowers and 1 white flower in the F2 generation, you might get your dominant ones being red, your recessive homozygous ones being white, but your heterozygous ones may be a shade of pink or somewhere in the middle like that. And that would be what's called incomplete dominance. Now, Mendel's principle of independent segregation, now he's talking about the alleles. Now, individual genes will sort independently into gametes. So, individual genes, so say your chromosome 1 and chromosome 2, they can, your genes, gene 1 and gene 2, don't necessarily always have to go into the same sperm or same egg. Right? So they can go into different ones. Now, each gamete receives 1 allele of each gene with a 50-50 chance. Okay, and we can use Punnett squares again to visualize the possible genotypes and phenotypes. And what we can see is that we see that genes will act independently to determine unrelated phenotypes. And this is important because we can have multiple genes making a single phenotype. Right? And that complicates things even more. So here's an example of a cat that is being mated with another cat. And what we see here is what's dominant is having a short tail. Okay, so short tail is dominant. That's why it's big S's. And brown is dominant. Okay. Now, when you're looking at multiple genes, right? So we're looking now here at 2 genes. It's the coat color and tail length. And each of these genes has a different allele. Okay, so you can be, for coat color, you can be white or brown. And for tail length, you can be short or long. All right? Now, remember this is parental. When you mate true breeding, meaning, once again, homozygous, parental generation, you're going to get all heterozygotes. And so all your heterozygotes, remember, are going to have the phenotype of the dominant allele. Well, the dominant allele, once again, is the dominant allele for coat color is brown. And the dominant allele for tail length is short. So, what we see here is that all of our F2 generation, all 4, are brown and have short tails. Okay? Now, you would mate this F1 generation to each other to produce your F2 genes, right? Your F2 offspring. And what we see here is that we're no longer looking at our 3-to-1 ratios because now we have 2 genes involved. So now we have 16 different possibilities. And what do we end up seeing? We see a 9-to-3-to-3-to-1 ratio in the F2 generation. And what is it? It's going to be 9 in the double dominant, right? So 9 are going to be brown and have short tails. 3, these next 3 are going to be one of the heterozygous looking ones, right? To where you're going to be dominant in one thing and recessive in another trait. So 3 are going to be brown with long tails. Now we have another separated group, the heterozygous group, right? Because they're independent segregation of these. So we're going to have 3 that are white and short. And then you're going to have 1 that is recessive for both traits. And that's going to be white and long. Okay? And if we find that one, here's the white and long. All right? And this is how we're going to be able to determine any of our true breeding lines where we're looking at 2 different genes. Okay, and we're always going to see this 9:3 to 3:1 ratio. Now, for an example, let's try this again. So using a Punnett square, draw out all possible genotypes and phenotypes of a cross between white flowers with long stems and red flowers with short stems, with white flowers and short stems both being dominant, and proceed all the way through the F2 generation. So remember, white flowers and short stems are dominant. Okay? And we have a cross between white flowers with long stems and red flowers with short stems. So let's write out, what are our parents? What do the parents look like? Well, we need one that's white. Okay, so let's give them the capital W's. We need it also to have long stems. Okay, so let's give them the small s's. Okay, well, now we need one that's red, so that's the little w's, and we need one with short stems, so that's the big s's. All right, so this is white and long. This is red and short. Okay, so if we mate these, what are we going to get? We're going to get the full heterozygote, right? So we are going to get, here are our gametes. I'll just write that out so we can see it. The gametes are only W and little s, and this is only little w and big S. So these crossed are going to give us only the, only this, right? So this is our F1. And what is that? What is this going to be? What's dominant? White's dominant, right? And what's dominant? Short. So all of these are going to be white with short stems. Okay. What are our possible gametes here? It's going to be WS, little w, big S, big W, little s, and little w, little s. All right? So now we're going to be able to figure out how to break down our Punnett square. All right? So here's going to be our F2. And so we draw these out, right? Because these are going to be the same for both maternal and paternal. So let's draw them out. So we have WS right there. WS over there. W little s, big W, okay, and this way, WS, we have little w, big S, and we have, okay, so now, okay, so let's go through and figure all these out. So this is going to be 2 big W's and 2 big S's. That's the easy one. And this is the easy one too, right? 2 little W's and 2 little S's. Those are the easy ones. Now we just have to fill them in. So this is big W, big S, little s. This is big W, big S, big S. Big W, big S, little s. Okay. This is, here we go. Big W, big S, little s. Big W, big S, big S. Big W, big S, little s. This is big W, little s, little s. This is big W, big S, little s. This is little w, big S, big S. This is big W, big S, little s. This is big W, little s, little s. And over here, big W, little s, little s. Little w, big S, little s. Oh, and over here, last one, little w, big S, little s. Now, if we want to look and see and prove to ourselves that this is in fact in a 9 to 3 to 3 to 1 ratio, let's pick out the simple one first. Okay. Here is our single one that has the homozygous recessive for both. So that's our 1. Okay. Now, let's find the 3 that have dominant in one and recessive in the other, and then the vice versa. So over here, let's see. We have, we'll go green for this. Okay. We will say, all right, this is dominant 1, not in the other. Dominant 1, not in the other. Dominant 1, not in the other. So this one is red and short. This one was red and long. Oh, sorry. This is red and short. Yeah, that one's red and short. Okay, good. And then what about this one? Okay. Now we're looking for the ones that are white. Right? We want to see the ones that are white. Let's see here. So we have white and long, white and long, and white and long. All right, so these are white and long. Now the other 9 are going to be the ones that are left. And those are going to be the white and short. And can we find those? 1, 2, 3, 4, 5, 6, 7, 8, 9. So each of these 9 have both a dominant allele for white, at least one for white and for short. Okay? All right, good. So let's go on to the next. So, as I said before, Mendel wasn't appreciated in his own time. And the only reason that he is now appreciated is because other scientists picked up his work and gave him credit for doing the initial work. So, in 1903, so this is about 20 years after Mendel died, the American biologist Walter S. Sutton linked cytological studies with Mendel's previous work. All right. Okay, this researcher, Sutton, emphasized the importance that diploid cells have 2 morphologically similar sets of chromosomes, and each haploid gamete received a single set. He stated that genes are found on chromosomes with 1 allele on each homologous chromosome. And we're going to get further into this as we move into the course. So to explain Mendel's work, Sutton stated that the gene for seed color was on a different chromosome than the gene for seed texture. Now, Mendel's second law states that genes should assort independently. But, as Sutton was noticing, many genes didn't behave that way. So these genes were said to be linked to each other. And what that means is that these 2 genes don't sort up independently at a normal 50-50 ratio. They're more often seen together than they are separated. Now, linkage occurs between genes because the genes are located within close proximity to each other and usually are on the same chromosome. Now, linkage is never 100% complete, okay, and that's due to the fact that you can have crossing over in meiosis, so in late prophase 1, but linkage can be above the normal 50-50, which would be considered an unlinked gene. Now, 7 years later, American biologist Thomas Hunt Morgan and his colleagues found a mutation in the eye color for Drosophila melanogaster. Now, this mutation was linked to the X chromosome. Right? Flies also have X and Y chromosomes. Now, the linkage of the white gene to a chromosome strongly supported Sutton's chromosomal theory of inheritance, proposed just 7 years earlier. Now, linkage to this sex chromosome was really apparent because the eye color of the progeny depended on the sex of the white-eyed parent. So the Y chromosome doesn't carry a copy of the white gene at all. So no white gene on the Y chromosome. All right. Now, for X-linked genes, the alleles expressed phenotypically by males, regardless of whether it's dominant or recessive, because it's the only copy. All right. So, an example here, we have this being the female, this being the male. All right, the male is white-eyed. The female is what we call wild type, meaning it's normal, okay, what we see in nature. And what we're seeing here is we have a white wild type female, a white-eyed male, and we have the F1 generation. Okay, this is parental. The F1 generation, both females, these are males, okay, both females and males are wild type, okay, regular brick red eyes is what they call them. Okay. Now, in the F2 generation, right, so these guys are going to be mated to each other. In the F2 generation, what we see is we get, this is a male, and these are two females. What we end up seeing is we get 3 that have the brick red wild type eyes, and 1 that has white eyes, and it is a male. Okay, so when the parent is with the white eyes is a male, in the F2 generation, the one that gets the white eyes is also going to be a male. All right. So the mutation for white eyes, we know this, when wild type is red, and we know that the mutation arose spontaneously but very infrequently. And what's important is that we know that males, which are, just like in humans, XY is a male, and female is XX. Okay? So males always donate their Y to any son, and they always donate their X to any daughter. And we know that sons always receive their X from their mom. Okay, so this is important. So using this information, let's find out what the F1 and F2 generations would look like if the original parents were a white-eyed female and a wild-type male. So the opposite of what we saw before. All right, so let's draw out our parental. All right, so we have a female that is white-eyed, all right, and we're going to call that little w. And we have a male that's red-eyed, okay, big W. And instead of putting another W there, remember, the Y chromosome doesn't have the gene for white on it, so we're just going to put Y for Y chromosome, all right? Now, when they mate, they're going to produce the F1 generation, which is what? What can we get? We're only going to get 1, right? Or no? No, it's different now. Okay, so we know the whole X and Y, right? So all of the females will get the X from their father. So they'll get a big white, right? So this is going to be a white-eyed, or sorry, a red eye, right? Let's make this clear. So this is red eye, and this is white-eyed. Okay, so the female is going to only be able to inherit that, right? So, and then they will get one of these from their mom. Okay, any males will be like this. The reason being, okay, and so what we see here is that the females in the F1 generation will all be wild type, right, which is red. And the males will all be mutant, which is white. Now, if we mate these two together, okay, what are our options? If we do our Punnett squares, Now, what we can see here is that we have 2 boys, 2 girls, so 2 females, 2 males. And what do we see? So let's look at this one. What is this one going to be? WW, okay? First of all, we know it doesn't have a Y, so it's going to be a female. And we see that it doesn't have a big W, so this is going to be white. Okay. All right, now let's look at this one. Okay, it's little w and y, so we know it's a male. And we know it doesn't have a big W, so it's also going to be white. All right, now we'll look at this one. Big W, little w. We know it doesn't have a Y, so it's a female. And we have a big W, so it's wild type or red-eyed. And then the last one here, big W, Y. Okay, it's got a Y, so we know it's a male. And what? We have a big W, so therefore it's also red. So that is what we see here. We get 2 white-eyed, 2 red, but we get both a white female and a white-eyed female and a white-eyed male. Good. Now, we can start moving closer and closer to current time, right? So now we're in the 19th century, or the 20th century, sorry, and 15 years or so after we were able to go through the chromosomal theory of inheritance, we can move on to thinking about how we can discover, or how we discovered, the genetic material of the cell. Now, to preface this, at the time, scientists believed that the genetic material of the cell were proteins. Okay, they didn't think that it was DNA, because at the time, the proposed structure of DNA did not lend itself toward replication. So they thought that protein was definitely the genetic material of the cell because it had enzymatic activity. So in 1928, we have British bacteriologist Frederick Griffith identifying what he termed the transforming principle, all right? So the genetic material of the cell. And he developed this extremely ingenious experiment that took this substance that's in a virulent Streptococcus pneumoniae, okay, a bacteria, and he was able to transform the non-virulent strain of this bacteria into a virulent strain. So virulent means is able to cause a disease. So how he did this was he took the non-virulent strain, or the R strain. Okay, it was rough looking when it was plated out on petri dishes, and he injected it into mice. Right, and then he took the blood of the mice, and there were no live bacteria. Okay, and the mouse was perfectly fine and alive. Then he took the smooth strain, the S strain, because it looked smooth. It had this extra protein capsule on it. And this was virulent, okay? And he knew this, and he injected it into the mice. All the mice died, and he was able to extract from the blood live Streptococcus pneumoniae. Okay, his next experiment, he heat killed this virulent strain. So he killed the bacteria, you know, kind of just like we do with viruses today, or sorry, with vaccines today. He killed it, but still injected it into the mouse. Well, the mouse lived, and he wasn't able to pull any live bacteria out of it. Now, this is the most important step right here. He took live R strain, which is non-virulent, and he took heat-killed or dead S strain. He mixed those together, injected into the mouse. And what did he find? Well, he found that the mouse died. And he found that he could recover live S strain. So he's saying, oh, hey, this has got to be the genetic material of the cell, is this transforming principle, right? That somehow was able to transform R and S. But what was that? We don't know. Okay, so fast forward a little longer, and this is kind of an aside before we jump back into the big picture, but in 1941, we have Beadle and Tatum proposing that genes direct the synthesis of enzymes. And they did this by irradiating spores on bread mold, and they found that these were unable to grow on media without arginine, which is an amino acid. And this amino acid was produced just by a single enzyme. So they came up with this hypothesis, oh hey, one gene makes us one enzyme. So one gene, one enzyme hypothesis. What I want us to understand is that this has later been amended to be a one gene, one polypeptide theory, because we can make more than one enzyme from a single gene, and that can be done by alternative splicing that we're going to talk about much later in this course. But that was kind of an aside. Now we'll jump back to the main story. Okay, so in 1944, now we have Avery, McLeod, and McCarty at the Rockefeller Institute showing what the transforming principle was. All right, now they say that it's DNA. And how did they do this? They took this heat-killed S strain. They fractionated it into DNA, RNA, and protein. Right? And each fraction was tested for their ability to transform the R strain into the S strain. Okay, so what they did was they separated into each fraction and only gave the DNA from the S strain to the R strain. Then they would do another sample where it was only the RNA from the S strain to the R and only the protein from the S to the R separately. Well, what they found is that the only DNA fraction that can transform an R strain to an S strain was the DNA. Okay? The RNA and the protein fraction did not. Okay? However, this was still not widely accepted because, as I said earlier, at the time, DNA's structure that was proposed by Phoebus Levine was not something that could be applied to being able to transfer information and be replicated. All right? Now, this is the reason why proteins were thought to be the genetic material. So Phoebus Levine, much earlier, so in 1910, he proposed what's called the tetranucleotide hypothesis. And he predicted that the structure of DNA was planar, and equal quantities of each nucleotide were in each plane. So it looked like this down here. So we had a guanine, a cytosine, a thymine, and an adenine in each plane. And they look like squares, and they added one on top of each other. Okay, so on and so forth. With the phosphate backbone in the middle and the bases on the outside. Okay, the bases on the outside. Now, luckily, due to this planar prediction, okay, where we had equal all four in the same plane all the way up, we had another researcher named Chargaff showing that this wasn't the case. So in 1949, we have an Austrian chemist, Erwin Chargaff, proposing that DNA of different species have different nucleotide composition, and they always follow this rule. The amounts of adenines always equal the amount of thymines, and the amounts of guanines always equal the amounts of cytosines. The amounts of A's and G's always equal the amounts of C's and T's. And importantly, A and T ratio, okay, A plus T, does not have to equal G plus C, but can be varying between different species. Now, this is important, and this is something that we know nowadays. We know that A base pairs with T and G base pairs with C. So therefore, this makes a lot of sense. At the time, nobody knew this. So back to discovering the genetic material of the cell, a few years after Chargaff kind of gave the light toward the end of the tunnel, we have these scientists named Hershey and Chase, these American bacteriologists and geneticists, conclusively demonstrating that DNA is a genetic material. Now, they did this in a really cool experiment. Now, they took bacteriophage. Okay, bacteriophage, same thing, is a type of virus that infects bacteria. All right? And the bacteriophage is very simple. Okay? All it's consisted of is DNA that's inside the head, and then the rest of it is all protein. Okay, so the head, the collar, the tail, all the outside stuff is protein, and only what's inside the head is DNA. So, what Hershey and Chase did is they took radioactive isotopes of sulfur and phosphorus and incorporated them into the growing bacteriophages. And you could specifically incorporate sulfur, okay, into protein. Okay, meaning the sulfur was only going to be in the protein coat on the outside. Okay. They also took the phosphorus, this isotope, that could be specifically incorporated into DNA, right? Meaning it's only inside the head group on that DNA. Now, what they did was, after growing the bacteriophages in these isotopes, they were allowed to infect the bacteria. And as we see here, we have our two different types, right? Ones that were just labeled with the sulfur. Ones that were just labeled with the sulfur. So only the protein. And ones that were just labeled with the phosphorus. So only the DNA. And what they see is you let them infect. So they latch on to the bacteria and shoot their DNA into the bacteria. And the whole point they want to do is make new viruses and break out. So they were allowed to infect the bacteria, and then they were thrown literally into this blender. And it's like a blender you would have on your counter. It is actually still held at Cold Spring Harbor in New York, the original blender that Hershey and Chase performed this experiment with. So they threw everything into this blender, hit blend, okay, and that basically shakes the bacteria off of everything, right? So it breaks everything open, shakes the phages, everything. And what they found is that the phosphorus, right, which we know is associated with DNA, was found to enter the bacteria as well as the progeny phage, but the S35, right, the sulfur that's in the protein coat, there was none inside the cell. It was only associated with the protein ghosts. That's what they're called, the ghosts. So it's the empty phage. So this was really great. So DNA is what's being passed on, not protein. So this is huge. In the same year, we have Rosalind Franklin. Okay? Rosalind Franklin is an English chemist and one of the premier X-ray crystallographers of her time. This, in fact, is a picture of hers called Photo 51. And this shows the diffraction pattern of DNA. And if you can actually look at it, we can see that we have a double helical nature, and we can even see the minor groove and the major groove between DNA. This is an amazing picture, especially at the time. Unfortunately for Rosalind Franklin, someone in her laboratory showed this picture, Photo 51, to some competing scientists without her authorization. And those competing scientists are very well known now. And the names of those competitors are James Watson and Francis Crick. So a year later, after getting that picture, that Photo 51 from Rosalind Franklin, they deduced the double helical structure of DNA. Now, they published their article in the Nature journal, and it was only 1 page long, and it's one of the highest, or if not the highest, cited paper from that journal. Now, what they said in their paper was that 2 DNA strands are held together by hydrogen bonds. And that's between the opposing strands, right? So the bases coming together make those hydrogen bonds. They said base pairing is specific. A pairs with T and G pairs with C only. And we've already talked about this. But at the time, that was a novel insight. Now, they said that the sequence of one strand defines the sequence of the other strand, meaning that it's complementary. And they also noted that it was anti-parallel. So remember, 5' down to 3' for one strand, 5' up to 3' for the other strand. And in one of their last sentences, they stated that the specific base pairing suggests a possible copying mechanism. Okay, so this was the whole basis of why DNA couldn't be the genetic material as seen beforehand, right? Because it couldn't be copied. But now they're saying, well, the phosphate backbone's on the outside, the bases are on the inside, hydrogen bonding to each other, they're complementary, and so therefore we could probably see a situation where you could copy this pretty easily, although they didn't propose the mechanism. So, as an example, based on Watson and Crick's solution, fill in the sequence on the bottom strand of DNA following what they said. So remember, A's pairs with T's, G's pair with C's, and DNA strands are anti-parallel. Okay, so remember, we have the 5' to 3' on the top strand. We have the 5' to 3' going the opposite way on the bottom strand. And if we want to make our bonds, T pairs with A pairs with T, C pairs with G, G pairs with C, C pairs with G. And let's not forget, how many bonds? A and T make 2 hydrogen bonds, A and T make 2 hydrogen bonds, G and C make 3 hydrogen bonds. And this would be our phosphate backbone right here. Of one strand, this is the phosphate backbone of another strand. So a few years after Watson and Crick deduced the structure of the DNA helix, we had an American biochemist named Arthur Kornberg that isolated this protein from a bacteria that he called DNA polymerase I. And he showed that DNTPs, or deoxyribonucleoside triphosphate, so it could be, they say that DNTPs meaning it could be an A, G, a T, or a C. He said that the DNTPs and a DNA template, okay, a DNA molecule, were required for DNA synthesis. And he showed using templates with different base compositions, so whether it's high AT or high GC ratio, he showed that the newly synthesized DNA molecules had a similar base composition. So confirming Watson and Crick saying that there was the complementarity between the two strands. And Kornberg established that a strand of DNA served as the template for DNA synthesis, but still he couldn't really offer the mechanism by which it happened. So there were 3 mechanisms that were offered at the time. Now we had semi-conservative, in which we have here, we start with our parent strand, all red, and the 2 new daughter helices, one of the strand would be completely parental and one of the strand would be completely new. We have conservative, where it's just like a photocopy mechanism. So of the 2 daughter strands, one would be completely old DNA, one would be completely new DNA. And then we have dispersive, in which case both of the daughter molecules would have new and old, okay, but we would have stretches where both strands were completely old and stretches where both strands had completely new DNA sequence. So how do we discover what type of mechanism this is? Well, 2 years later, we have Messelson and Stahl showing that DNA replication is semi-conservative, meaning you have the original DNA, all right? When it is remade, we have one strand being completely old and one strand being completely new. Now, how did they do this? Well, it's a really, really ingenious experiment. DNA was made denser or heavy by adding the heavy isotope of nitrogen, so nitrogen 15. And then it was subjected to high-speed centrifugation through this dense medium of cesium chloride. Okay, so you either grew DNA in the what's called light nitrogen, so the normal N14, or the heavy nitrogen, the N15. So you called that heavy or light DNA. Right? So they grew that through 14 generations, right? So that there was no bias coming in. They were all different, right? Or sorry, you either had completely N15 or completely N14 was what it was showing. So after one round of DNA replication, in the presence of the light isotopes, all of the daughter molecules had an intermediate density, or heavy light. So from 100% heavy to 100% intermediate. And then what they did is they did one more replication. At that point, they saw a reduction of the medium, the intermediate, down to 50%. And then they saw 50% being fully light. Okay? Now, this demonstrated that DNA replication is semi-conservative because you were having the old parental strand being the heavy, right? And then you grew it only in heavy to start with. Then you started growing it in light. So you could only be adding light, right? You couldn't add more heavy. So by going from completely heavy to intermediate after one replication, that's saying, well, it has to be half and half, right? Half old, half new. Well, this doesn't necessarily mean that, so right there, that breaks away conservative, right? So, but we could still have distributive, right? Or dispersive, as well as semi-conservative. So what they did is, they did more and more generations and found that as you increase the generations, you're not adding any more of that old stuff, right? So you're not distributing it in between strands. It's every new strand is going to be a light strand. So that's how they could figure that it was semi-conservative replication. And that's a big, big deal here. So that's the end of our DNA, right? The discovery of the genetic material and how it is replicated. So this is a quick overview on the discovery of RNA. We'll talk more about RNA later. But ribosomal RNA is one of the three types of RNA. All right, so our RNA was discovered in the 50s by Albert Claude and George Palade. Transfer RNA, so ribosomal RNA is important for the structure of ribosomes, as well as it serves just a structural purpose. Transfer RNA is what is responsible for bringing in amino acids to the ribosome to match up with the particular anticodon on the mRNA. So transfer RNA was found in 1956 by Zemechnik and Hoagland. And then messenger RNA, which is responsible for, which is made from DNA and is responsible for dictating the sequence of amino acids for protein synthesis at the ribosome, was discovered in 1960 by Brenner, once again our friend Francis Crick, as well as Jacob and Monod. And now that we've learned about our DNA and RNA, we should talk quickly about the central dogma of molecular biology. And what does that really mean? And this is something that we're going to talk about many times throughout this course, but the simplest way to explain it is that DNA can make more DNA, right? And that is called the process of replication. Right? DNA can be turned into RNA, and this is the metabolic process of transcription. And finally, RNA can be turned into protein via the process of translation. So this is what everything that we believe in molecular biology follows. Now, one thing that violates the central dogma is something that you might be familiar with that's called reverse transcription. Now, this is turning RNA back into DNA, and we see this in the type of enzymes such as retroviruses. So they have a sort of enzyme called reverse transcriptase, and that is able to turn RNA back into DNA. So this is an important thing to always have in mind when we're thinking about the molecular biology central dogma. So RNA can be made into protein. Well, how do we figure that out? And how do we figure out which RNA will turn into which amino acids? Well, cracking the genetic code. Now, this is important. If we remember the genetic code, or if we haven't learned yet, the genetic code is what tells us what triplet repeat found in an mRNA will give us a specific amino acid. Now, in 1961, we have an American biochemist and geneticist named Marshall Nirenberg. He discovered the first DNA triplet that would go on to make an amino acid. And he did this by taking a bunch of nucleic acids, so RNA, that only was made up of U's. Okay? And he fed this through the ribosome, in a cell extract, and only phenylalanine amino acids were made. So he found out that the first triplet codon ever distinguished was UUU that coded for phenylalanine. Now, it took about 5 more years, and some other researchers helped, but the code was completely finished in 1966, and what they found is that there were 64 possibilities, okay, of triplet repeats, okay, or triplet codons, sorry. And each one of these could code for an amino acid, or so they thought. 3 of them they found, and these are important, were found to not actually add an amino acid and instead cause the release of the polypeptide chain from the ribosome. But the other 61, okay, the 61 other than these 3, the 61 possible codons all coded for amino acids. And what's also very important is that, for example, let's look at this. 4 of these different codons will code for the serine amino acid. And this is what we call degeneracy of the genetic code, meaning that we could get the same amino acid from multiple different mRNA bases, meaning also, since we got the mRNA from the DNA, we can have different genetic sequences giving us the same amino acid sequences. Well, I hope you enjoyed the lesson. Thank you very much for coming, and please come back to educator.com.